Speaker is Huimin Cheng, who is also at the School of Public Health in Biostatistics. So, Huimin.
Okay, good morning, everyone. I'm so excited to be here to share my recent work. Actually, it's still an ongoing work, knowledge graph and drug repurposing. So we often hear about breakthrough drug, but do you know only 5% diseases, they have FDA-approved drugs. So that means 95% of diseases that don't have FDA-approved drugs. So that means millions of patients in the world, they are left with no treatment. Why is this happening? This is because developing a new drug is so time-consuming and so expensive. So on average, it takes us 12 to 15 years and cost us $2 to $3 billion to develop a new drug. We don't want to wait 15 years and we don't want to cost $1 billion. So instead of developing a new drug, maybe we can think about finding new uses for an existing drug. So that's an idea of drug repurposing. There are some successful examples of drug repurposing. One famous example is minoxidil. So minoxidil is a drug which was, originally developed to treat high blood pressure. And when this drug comes out, patients take this medicine, and then patients find something unexpected. Their hair is growing back. So they're not only getting, better blood pressure, they're also getting a better selfie. So then, scientists find, okay, this drug may be repurposed to treat, hair loss. So that is a successful drug repurposing example. And this example is actually very lucky. But we cannot depend on luck every time, right? We want to predict the new uses of some drug before they surprise us. So that's why we need computational way. In this way, the knowledge graph plays a very important role. The knowledge graph is like a giant map of biomedical knowledge. Each node can represent a biomedical entity. It can be a drug, it can be a disease, it can be a gene. And we also map the relation between these biomedical entities, like a drug can treat a disease. So we draw a connection between this drug and this disease. So that's knowledge graph. So knowledge graph is very powerful, but how do we construct this knowledge graph? We usually have two types of ways. One way is knowledge they're buried in massive literature, and every day we have like thousands of papers published, so we can get most updated information from the literature. And another way is there are some existing database like Drug Bank, DisGenet, and CAG. This database, they have some curated, verified relationships in this database. But the advantage of using the literature is we can get the most up-to-date information. For example, when a new disease like COVID comes, we can see immediate information in the literature, but in the existing database, it takes some time to update the verified relationships. So in this talk, we focus on extracting knowledge graph from the literature. This way is not easy because there are so many literature, so many papers. So for example, if I search COVID in PubMed, I got 300,000 results. So that means if I take 30 minutes to read a paper to extract knowledge, it will take me 19 years to read all those papers, and to build a knowledge graph, it cost me 19 years without eating, without sleeping. That's basically impossible, right? My life is so short. I don't want to spend 90 years in this. So, but fortunately now we are in an AI age. We have ChatGPT, we have large language model. We can definitely borrow strengths from this. So, here's an example. This is an abstract of a PubMed paper. I feed this to ChatGPT. I ask GPT, can you extract the biomedical entities in this paper? And GPT returned these results. So at first look, GPT did a very good job the GPT extracts COVID-19, ACE2, those entities from the abstract. But when we take a closer look, we compare the GPT results with our manually labeled results, we find something unsatisfactory. For example, there are two pathways, the RAS and the mTOR signaling pathway. These two pathways, they're not even mentioned in the abstract, but they are identified by GPT. GPT infers too much, so GPT generates some hallucination. So we need a better AI model. That's why we develop a reinforcement learning enhanced LLM. So the idea is we choose like 1,000 papers and let the LLM get some results, and then we hire some domain experts like master student or PhD student in the medicine area, and then we ask these domain experts to evaluate the results of the LLM, and given the feedback, we train our LLM to let the LLM to learn from its mistakes. So after this kind of iteration, we get a better results. And here's our extracted knowledge graph from that abstract. Except for extracting the biomedical entity, we also need to build the relationships. And here we define 21 different types of relation between those biomedical entities. For example, a drug can treat a disease, and for example, biomarker. Gene can be a biomarker for a disease. And in this knowledge graph example, I use different color to represent different biomedical entity. And here we can see SARS-CoV-2 causes COVID-19, and COVID-19 affects two tracts. Like the first is respiratory tract, another is the gastrointestinal tract. So this knowledge graph gives us maps the information from the abstract to a structured graph. So that's how we construct the knowledge graph from the literature. After we construct the knowledge graph, then we just fit into a graph neural network, GNN, and we use it to basically do the link prediction task. We predict the probability of a drug can treat a disease, and our output is a probability. So let's see a small-scale case study. This is a case study. We use the COVID-19 related PubMed, and the PubMed, they are published in January 2020 and April 2020. Why do we use this small-scale example instead of showing the large-scale knowledge graph by combining all the literature? Because we are still processing it. There are tens of millions of publications, so it takes a lot of time to process that. So in this presentation, let's just see this small-scale example. So we feed this knowledge graph from these publications to the GNN and get some prediction. And our top prediction is the hydroxychloroquine and neck cancer. So before the May 2020, we used the literature, before May 2020 to make predictions. And before that time, we do not see any literature mentioning the hydroxychloroquine can be used to boost the treatment of neck cancer. But our knowledge graph can predict this. But how do we validate our prediction is correct or not? We use a scientific kind of like a future view. We use the publication published after May 2020. So if we find this relation really show in the future publications, we cannot say, okay, this is correct. And very exciting, we do find a paper published in May 2022 that do verify hydroxychloroquine can boost the treatment of neck cancer. So in this way, we can calculate some accuracy. For the top 10 predictions, we calculate the accuracy, which is the number of validated prediction over the number of predictions. And top 10 predictions, the accuracy is 100%, and we can also consider top 16. And when we consider top 16 predictions, the accuracy goes down, but it is still over 80%. So that's the small scale example. But I have to admit, this work, we still have a lot of work to do. And the first thing is we need to combine all the literature to build a super large knowledge graph. And the second thing is, after constructing the literature-based knowledge graph, we will combine it with existing database like Drug Bank, CAG, because they do have some verified relationships. So we will combine them together to build a more comprehensive knowledge graph. So stay tuned. Thank you very much.